Thank you all for coming to hear about some research that's been of interest to me in my lab for some time now. Today we're going to be talking about the development of a high-quality genome for the genus Lucena and the use of that genome and other resources to understand some important duplication events that predated the evolution of the genus Lucena. Before we get into talking too heavily about Lucena in particular, it's worth reminding all of us that the legume family is now classified into a 6 subfamily system, and that the traditional mimosoid subfamily is now a member of the C-Salpinioideae, and Lucena is a member of the C-Salpinioideae. It's also worth mentioning that genomic resources for legumes are not really lacking. Well, they are lacking, but they're relatively well developed compared to some families, but that the majority of those resources are from economically important papilionoids, and that there's plenty of room for the addition of more critical and well-dispersed genomes across the legume family for future research in a variety of areas. Now, there's been considerable both interest and activity in using phylogenomic resources from a variety of data sets to try to understand patterns of diversification in this family. This is a slide from Kuhn et al. In 2020, where they were investigating, in particular, the pattern of divergence among subfamilies and deep branches within this family. And from a very comprehensive analysis and exceptional data set, they concluded that there really is a hard polytomy down at the base of the legume family, and that there really isn't an obvious way to decipher the pattern of diversification in the group. One of the contributing factors to the complexity that's observed at the base of the legume tree is whole genome duplication. Now, this is another paper by Kuhn et al. In 2021, in which they were looking at both the timing and placement of whole genome duplications down toward the base of the tree. One important diversification was inferred along the stem lineage prior to the diversification of all the subfamilies. And then there were at least 2 other whole genome duplications along stem lineages to subfamilies, and that some of these may have actually been allopolyploid in their origin. Now, I bring this up just to point out that the legumes are impacted by whole genome duplication in ways that many plant families are, but they're characteristic of this family. They not only occur here at these deep nodes, which have been a focus of recent research, but also out in the more terminal branches of the tree. And many of us will be familiar with, for example, soybean as an example of a branch of a legume tree that's been affected by polyploidy. Another one of those areas is this genus Lucena that we're focusing on here. This is just a slide showing you some of the interesting morphology that can be observed in pairwise comparisons of a few different species. There's 24 species in this particular genus, and they occur naturally from Texas down into northern South America. There's also been interest in this group because of a variety of uses these plants have had, both historically in their native range for human food use, and in modern times, particularly around the tropics and subtropics as cattle fodder, soil amendments, biofuels, and other sorts of areas. Our current understanding of, or the present published understanding at least, of the diversification of this lineage in the genus Lucena comes from my lab in 2011, where we hypothesized that, you can see my mouse here, there was a polyploidization event along the stem lineage, excessive and extensive diploidization, leading to what might be referred to as a diploid speciation, sort of diploid radiation, that we inferred was largely allopatric in its origin, but that some of these lineages then came back together to form a series of tetraploids, all of which were allopolyploids. And we hypothesized that many, if not all of these, were the result of human translocation of seeds into backyard gardens where these hybridization events occurred. So one of the things we're interested in is trying to continue to develop our understanding of this particular pattern or these series of hypotheses in the group, and particularly things like, is there really a paleopolyploidization event here? Has there been extensive diploidization, etc.? And then later on in the future, we'll be looking more and more at other elements deeper in this tree. So for objectives today, we're interested in investigating this whole genome duplication that predates the divergence of Lucena. We're also interested in developing a reference quality genome for mimosoids, in particular, developing one for Lucena so that it's available for investigations among its close relatives. And then ultimately, we'd like to use the genome and other ancillary resources to look at the fate of genes and gene families that come from this whole genome duplication event, or any whole genome duplication that predates Lucena. So the first step I'm going to talk about then is the genome assembly. And there's so much more that can be said about the genome assembly, but in short, I'll say that this particular version comes from the availability of 50xpac biocoverage, where we used a Canoe assembler to generate what we'll call here as draft genome 1. It's really probably draft genome 1000. Not really, but draft genome 50 is more like it. This was an 816 megabase assembly, moderately well assembled especially at the time. 7500 contrigs was quite good, and NM50 of 232 kilobases was wonderful, but still not as well assembled as we would like. We were fortunate to be able to move on to incorporate Hi-C data and genome polishing from existing Illumina data we had from previous versions of the genome. This work was done by Phase Genomics. And here we're able to move on to a much higher quality genome with only 118 molecules. So the scaffolding worked beautifully. And N50 of 30.8 megabase pairs. We took that genome, and Susie Strickler from Boyce Thompson Institute worked with us on annotations. And so the final assembly that we've got here actually ends up looking more like this. We've got 28 pseudochromosomes, corresponds to what we would have expected in this group. We've got only 90 unassembled contigs. We've got some other characteristics I'll talk about in a minute. And the annotations included about 55,000 gene models. So now what we want to do is take a look at how this Lucena assembly may compare to some of the other well-developed legume genomic resources. And so just as a quick assessment of some of those features to what quality this genome might be. Well, first, this doesn't necessarily reflect quality, but you see that Lucena is on the sort of medium to higher end of typical legume genome sizes, or genome assemblies, I should say, at this point. The number of scaffolds or contigs in the assembly is really very small, and that's quite a good sign, given that it's a fairly robust assembly. The mean scaffold size and contig length is very important, and here Lucena is doing extremely well. This is not only a function of having pseudochromosomes, but also a function of having assembled most of the pieces into those pseudochromosomes and not having lots of unassigned contigs. Gaps in the assembly are also something that people often look at for the quality of assembly, and Lucena is on the low end in terms of number of gaps in the assembly. So what it appears here is that we have a well-assembled, highly contiguous genome. Now, how does that look when it comes to gene space? Well, we first looked at this from the genomic component. So here we have Lucena. We can see here the Lucena tricandra assembly. The blue colors across here represent so-called complete BUSCO genes. So these are an estimate of how well assembled the genome is. We've got about 98% complete BUSCO genes here, and very few fragmented or missing BUSCOs. And if we look down at all the other legume genomes, generally speaking, these genomes are quite good in regard to the genomic assessment of their BUSCOs. If we move on to look at the results of their protein annotations, we see much more variability. And this indicates that some of these genomes could probably use better annotations in general. But if we look at Lucena tricandra, you'll see here again, now we're at about 97% BUSCO completeness when it comes to the annotations. And this is on par with soybean and phaseolus. And so actually, it really looks to be among the top 4 in terms of completeness. Very nice complete genome. Very nice complete genome. So Lucena, the basic take-home message here is we have a nice, very well-assembled, contiguous genome that also captures gene space in a way that relatively few of the other legume genomes do. So it's a high-quality assembly. Very useful, hopefully, in the future. Now, I want to go back and just mention that some of you may have noticed there's 2 different colors of so-called complete BUSCO genes. And the pale indicates the single copy, the genes that were present in single copy, and the blue indicates those that were duplicated. And you'll notice that Lucena has a high level of duplication, as does Prosopis and as does soybean. And we know soybean is now a tetraploid, or a paleopolyploid, I should say. And so we come back to our figure here and ask, what have we learned from the genome that's of interest? And one is that this idea of heavy diploidization following the paleopolyploidization event, wherever that event is probably not as complete as we thought. It's clearly not as complete as we thought. So perhaps we should, scale that diploidization down quite a bit and say, there was some diploidization along here, but this thing is really behaving more as a tetraploid, perhaps. or somewhere between a diploid and a tetraploid than we might have predicted previously. Now, our next question we're interested in is, what about this paleopolyploidization event? There's a lot more data out there now, better ways of looking at where, rather than just chromosome number, where does this duplication occur? So here we enter Jens Ringelberg, from Colin Hughes's lab at the University of Zurich. And he first used a no-tongue analysis to try to identify where, there are more species in this tree than are shown here, where possible duplications might have occurred among Lucena and its close relatives. And one possibility was that there were two parallel whole genome duplications, one for Lucena and one for Schlittia, or the other possibility is that there's one that subtends the entire group. So he went on to then actually test this using whole gene WGD-GC analyses, and he looked at a series of nested analyses and concluded fundamentally that the whole genome history is best characterized by two independent duplication events. So Lucena does indeed appear to have its own unique whole genome duplication events somewhere along its stem lineage here. Now we're in the process still of trying to date that more precisely, and for that Eric Kunin, also from Colin's lab, has been working on these elements. We're not quite finished with that portion where we're using duplicate genes specifically from Lucena to precisely try to date that duplication along the stem lineage. But what Eric has done before, or has done previously, and it's available now for us, is to look at or to calculate the crown ages and the stem ages across the mimosoids, across mimosoid groups. And this is what he sees for Lucena. And if we compare the two means for the stem and crown, presumably the duplication event occurred between these two. And so the duplication is somewhere between 14 and a half and 20 million years ago, or 21 million years ago. So what we're learning in this particular case is that we have a high quality and highly contiguous genome for Lucena, so that's great. The genome retains much more duplication than we had thought before. So this is perhaps better a paleotetraploid rather than being referred to as a diploid. The results of the analysis to look at the position of the whole genome duplication that predates the evolution of the genus support an event both along the stem lineage to Lucena and also one to Schlittia. And the whole genome duplication event along the stem lineage to Lucena likely occurred between 14 and 20 million years ago. I'd hoped to be able to present more about the fate of individual of specific gene families in Lucena, but we're still working with that, and hopefully that'll be done in the next relatively short time frame, and we'll get this genome and these analyses out in form of publications relatively soon. I'd like to just mention that there are a number of people who've gone into helping work with Lucena genomic resources. The genome was particularly initiated by Plant Genome Research at NSF. I will leave this slide up and go ahead and pause there for questions. So thank you all for being here, and I'm happy to take questions.